Okay. All right. So we've got a bunch of people joining. Thank you guys for joining us tonight. If we haven't met yet, my name is Melissa with Pura Vida Divers. I run all of our events and coordinate all of the fun things for you guys to see and do with us here. So this month we are really excited. We're focusing all on coral reefs this month, and it ties in really well with our last month's feature, which was ocean conservation. So we focused a lot on the removal of marine debris from our reefs and our wrecks offshore and from local beaches. And then this month, we're really excited to learn a lot more about coral reefs, what they are, why they're important, and to continue our cleanup efforts offshore and on local beaches. So real quick, we've got a couple really interesting activities all set for this month. So we have a coral reef conservation class. That's actually a PADI specialty certification class. We're hosting that at 6:30 p.m. on the 27th. It's a Tuesday night. And it's actually a PADI cert that you can tune into online, and you can learn all about coral reefs from home, and you can actually earn your specialty certification. You don't even have to get off the couch. You can do it right from there. And then this month, just like last month, we have all of the cleanup supplies you need, both at the boats and here at the shop. If you'd like to swing by, we'll provide you with all of the materials you need to go do a cleanup here on the beach, at Blue Heron Bridge, or offshore on the boats. So we're super pumped about that. We've already removed a lot of trash, and I'm excited for us to remove even more. And we do report that both through the Dive Against Debris app, if you are a Dive Against Debris diver, and through the Ocean Conservancy's Clean Swell app as well. So with all of those exciting events soon to come, I am going to pass it over to Dr. Joana Figueiredo. She is with the Nova Southeastern University, and she is a coral reef researcher. So Dr. Joana, let me pull you up.
All right, hi everyone. All right, thank you everyone for coming. So I'm Joana Figueiredo. I'm an associate professor at Nova Southeastern University. And as they've mentioned, I am been working with corals now for a while, trying to induce corals to spawn. So just to start our presentation, you know that coral reefs occur throughout the world, and so in very different places, but always in the tropics. And I really like this map because I think it's almost like a nice bucket list for any diver of all the places we want to go. So they are all over the world in the tropic areas, and obviously they can look very different from place to place. So this is a reef here in the Florida Keys, which is quite different from some reefs, for instance, in the Pacific, where you have a bit of more higher diversity of corals, but then less octocorals. So it's a different type of environment. And if you go a little bit deeper, you find also that you find other species of corals that are a bit more typical from deeper areas. In some areas, the reefs do not have a lot of corals, and so you see on this here, there's a coral here, another staghorn coral here, another one there. In other places, the floor is completely covered in corals, and there's hardly any free space available for anything else to settle on. And corals can have many different shapes. So now you can have the sort of plated corals and the branching ones and the bouldering ones, and it's kind of like more leafy-like corals. But they all are made of repetitions of polyps, as you can see here. So you have a little polyp, and then that polyp will just make copies of itself and create a colony. But each polyp has its own mouth, its own stomach, and then it has tentacles. And these tentacles are used to grab food that they can then put on their mouth, but also to sting other corals. So I guess a lot of times people don't associate corals with being very active animals, but in fact, they can have a very active life. So if you put two corals together, or if two corals start growing together, they actually arm each other, and they throw these tentacles, and they sting each other and actually kill the tissue of the other coral that is trying to grow next to them. So it's actually, if you go around on the reefs in some places in the Pacific where there's hardly any free space, you see things like this, where you have corals that do not allow. So for instance, this round coral here you see in the center is a very aggressive coral, and it does not allow this other one, just a plated one, and a cropra, to grow on top of it. So it just stings it and doesn't allow, so it creates these marks here around it. This one is trying to grow around the other one underneath, and but it has to keep a certain distance, otherwise the other one will again sting it to that. And then also you see this sort of the plated corals that also grow very fast relative to this other one, round one, but the round one is more aggressive and therefore can still make sure that it's not covered on top by the other coral. So yes, they work, they move, it's just that as much, the temporal scale that they act is just much longer. And in terms of reproduction, and in terms of sexual reproduction, corals can have two modes of reproduction. They can be broadcast spawners, meaning that they release eggs and sperm into the water column, and then the sperm and the egg will get together and fertilize, and then do all the embryogenesis in the water column, and then until it creates a larva that is swimming. And actually, the larva is looks kind of like a pear shape. It's very small, obviously. It's a pear-shaped larva that has like a fuzzy air around it, almost like a peach, but it's still enough that can allow it to swim around. And then what the larvae does is kind of like pokes the bottom, and depending on what smells it has and whatever it finds on the bottom, it determines if this is a good place to settle on, because once they settle, they'll have to live there, they'll be stuck there forever. So they need to find a good place. So they usually are looking for places that have crostose coralline algae, so that pink encrusting algae that we see on the reefs, on healthier reefs. That usually is a good indication that it's a good place for corals to grow. And also sometimes the smells of other corals also are indicative of a good spot to just settle and go ahead and do metamorphosis. So that's what the larva does. When it reaches to the bottom, it goes into a metamorphosis and it turns into a polyp with tentacles. And then that polyp will start making copies of itself, like clones really, and then it grows into a colony. The other way of reproduction is the brooders. So the brooders have, what they do is they release sperm into the water, then the sperm enters into female colonies, and then the fertilization occurs inside the female, and the larva develops still inside the coral, and then when it releases a larva that is already swimming and ready to settle immediately. So there's these two strategies. Most of the corals around the world are broadcast spawners. Here in the Atlantic, there's definitely a much more higher number of brooders than in the rest of the world. And in terms of sex also, you can have species where some individuals are in this species, some are males and other ones are females. As you can see in this example, on the left, you have a male releasing sperm, and on the right, you have a female releasing eggs. But most of the corals actually are hermaphrodite, and this is what they release. It's a bundle of eggs and sperm. So what you see there, the orange things are the eggs, and the white parts are just sacs of sperm that after a while of being in water, this bundle just breaks, and then this eggs and sperm are released into the water. So most corals are actually hermaphrodite. They can work as, they have inside each polyp, they work as male and female. Another very cool thing about corals is that corals have this very interesting symbiosis with a unicellular algae called Zooxanthellae. This algae actually are what gives color to the corals. If corals did not have the symbiosis, their tissue would be transparent, and what you would see, they would look white because what you would be seeing is their white skeleton underneath. So this algae, the algae is when you go to a reef and you see these corals that are, pink and blue and green and orange and all of that, that's actually the algae that lives on their tissue. And the cool thing about this algae is obviously not that it just gives color to the coral, but it actually does photosynthesis. And so it uses some of the byproducts of the digestion of the coral, but because the coral is eating zooplankton and phytoplankton, and it uses some of those byproducts and then light to do photosynthesis, and then it provides also that food to the coral. So the coral actually has two sources of food. So the plankton, and then this algae that live on, and them, the products of their photosynthesis. And that is, I wanted to introduce you that idea about the symbiosis, because this is going to be important to explain why corals are declining worldwide. There's a big decrease in the coral reef ecosystems around the world. The number of corals are decreasing, the size of these corals are decreasing. So here on the left side, you just have a graph and looking into how the percentage of coral cover in the Caribbean throughout the years has been decreasing dramatically. But it's not exclusive to the Caribbean, it's happening throughout the world. And just 2 or 3 days ago, the Great Barrier Reef has, trying to move this in that area, the Great Barrier Reef has just, researchers there have just released this paper that is showing that the Great Barrier Reef has lost about half of their corals. Their corals are now smaller and less of them. So it's becoming a problem everywhere. And that is not exclusive to the Great Barrier Reef and to just the Caribbean in general. It's also in our area here. So this is for the Southeast Florida, so all the way up to from Miami-Dade up to Point St. Lucie, and you see that the percent of the floor of the bottom that is covered by corals has just been decreasing. It's about half that it was just even 10 years ago. So this is just very dramatic. And what's causing this decline is climate change. So mostly the warming of the water, pollution in the way of nitrates and phosphates that come out with fertilizers, and overfishing. And so particularly what's happening is, when we put fertilizers in our lawns to make our lawns nice and green, we're also really, those same fertilizers are also fertilizers for algae, right? And algae then take over space that is usually occupied by corals and grow on top of corals. And then when we overfish, particularly fish that eat algae, then we don't have those fish there controlling the amount of algae we have on a reef, and then it makes just the problem worse. But I have to be honest in saying that the worst issue that we have in coral reefs worldwide, because it even affects reefs that are completely away from human populations, is this warming. And what's been happening is, with the increase in carbon dioxide in the atmosphere since the Industrial Revolution, we have been seeing an increase in temperature too. And corals evolved in a very, very stable environment where temperature doesn't change too much. So even 1 degree, 2 degrees above their summer temperature is enough to get them stressed out. And when they get stressed out, what they do is they release that algae that lives on their tissues that gives it that beautiful color. And therefore, and what we say that it happens is that they bleach. So they pretty much, they look all white, and that's why they say bleach, because they look completely white. So bleaching is a stress response. It can happen when it's too warm, when it's too cold, when there's pollutants in the water, when there's a lot of fresh water. So it's a stress response. But the cause that we're seeing so much bleaching around the world is mostly warming. And what happens is you go from a very healthy reef that is nice and colorful to this bleached reef, and the coral, when it's bleached, it can still be alive for a while, but because it does not have a lot of energy reserves, because it's only feeding through, feeding on phytoplankton and zooplankton, then over time it will start not having enough energy to make repairs of lesions and things like that. So it ends up, if it stays on this condition for a long time without reacquiring this algae, it ends up dying and then getting completely covered in microalgae. So one of the processes, well, let's put it like that, the corals have evolved in tropical areas, and tropical areas are known, as you live in Florida, in areas that have hurricanes, there's a big severe storms that's often, disturb a reef and just kill a reef, for instance, throw sediment on top of the reef and things die. But they have naturally they have processes that allow them to recover from this. So if you think about that, each of these green dots is a reef, you can think that as they reproduce and produce their larvae, that some of this larvae will settle back onto the same reef, repopulating it, but also can send, because the larvae need to spend some time in the water, they can actually disperse and go to another reef and then for repopulated. So if you have a whole system where all the reefs are healthy, and then a disturbance comes and actually kills some of the reefs that now you're in white, the ones who are still healthy will be able to produce larvae that then are dispersed to these reefs that have been disturbed. And over time, you should be able to get a system back into the original state. So they have systems and mechanisms to recover after disturbances and recover after small bleaching events. The problem that we're having is that now they're not recovering. And they're not recovering because in general, what we're finding is that they're not being able to produce enough offspring that can replace the adults that have died. So usually what you have is males release sperm into the water, and then females release the eggs. And because they're so close together, there's a high density of corals in this area. The eggs always meet the sperm in the water and therefore create, get fertilized, create embryos that then develop into larvae that then can settle in the new environments. But at the moment, because there's been so much coral dying, now these corals are so far apart from each other that even if they're still reproducing, which it can happen, the eggs and sperms from different colony may never meet, and therefore they never produce babies. And then it just makes it even harder to get these reefs to recover and to replenish themselves. So the solutions obviously need to be reduction of greenhouse gas emissions and protection in a way of minimizing or eliminating direct local impacts such as pollution and overharvesting. But and these things are absolutely necessary, and without them, we cannot protect coral reefs from further destruction. But we can, one of the things we can do to at least accelerate the recovery alone by itself is not sufficient, but that can accelerate recovery is restoration. So one of the things we can do is try to increase the coral density on the reef by growing corals in captivity or in offshore nurseries, and then planting them on a reef, epoxying these baby corals on the reef. And the way we've been doing this has been mostly through processes of asexual reproduction. So pretty much we go to a coral and we cut a piece of it, and then we just glue that piece on an area that has been disturbed so that that coral can repopulate this area. And these are just purely clones, so they are exactly the same DNA, right, the same individual. But what we want to start doing more is sexual reproduction. And the whole advantage of this is that we are creating new genotypes, new individuals. Like humans, it's a recombination from the genotype of the mother and genotype of the father, and they'll get just a completely new individual. And actually all the babies from this cross between a male and a female will be all completely different from each other. And by having such diverse offspring, it may mean that some of them may be, for instance, a little bit more tolerant to a little bit warmer temperatures, a little bit more tolerant to a little bit higher turbidity and things like that. So it is an important tool to use sexual reproduction. So the cool thing about corals is that they actually have a very long period for their gonads to mature. So females are pregnant for around 9 to 11 months. You can see here on the left side, this is an acropora coral, staghorn, from our area. And this was, the branch was just broken right before the time of the full moon when they spawn. So you actually can already see here the eggs inside the polyps. So they take about, as I said, 9 to 11 months to make the eggs, and about 3 to 4 months before the time that they're supposed to spawn to make the sperm. So sperm is a bit cheaper, and so they just do it a little bit later on. It doesn't have as much energy content. So as the corals in our area here in Florida, they spawn August or September. That's usually the main times they spawn. So actually by November, December, you should start seeing the females laying their first eggs. Obviously the eggs then take a long time to mature until August or September of next year. Coral spawning, though, only happens once in a year. So it's a once in a year event, and it's really cool, and it's very, very, very synchronized. So I guess the question that a lot of people ask me is, why do they synchronize it? Why is it that they all synchronize the spawning? Well, within a species, it's good if they synchronize it. It means that the eggs and sperm of the same species will meet and therefore facilitate fertilization. But between different species, why is it that they all spawn around more or less the same time? And the reason for this is that if you have a lot of eggs and sperm on water, particularly the eggs, which are energetically very good for a fish to eat, if there's so much food available, there's only so much each fish can eat. So the individual risk of each egg to be eaten is reduced. So the fish can eat a few eggs, but they cannot eat the whole, all the eggs that each coral produces because there's so much available. And in these periods of coral spawning, there's actually a lot of not just corals are spawning. A lot of other animals that live on the reefs decide that this is a good time for them to spawn to for the same reason, trying to avoid predators. So you see Christmas tree worms and sea urchins and all these other animals, snails, worms, everything spawning at the same time. So in a place that has a really high coral cover, it can be a very interesting place to be on a reef when they spawn, and actually can be pretty smelling. So, but how do they do it? How do these corals come up with a specific day, specific hour to spawn? So it's based on cycles of the sun, the moon, and the earth, and the relationship between them. So in terms of the month that they spawn, it seems to be related to the annual temperature cycle. So if you see here on this graph, we have several years. This is actually for the Great Barrier Reef. So the temperature is actually high because it's the southern hemisphere. So actually they are the coolest in June, July, and then they're warmer in January, February. And they usually spawn here in October, November. That's usually when they spawn. And actually, it seems to coincide with the fastest rate of increase in sea surface temperature. So that's their cue for then the release. But they start producing the eggs again, the 9 to 11 months before. In terms of the exact day and time, seems to be more regulated by cycles of the sun and the moon, and actually the relationship between them. So for instance, is known for most species that they will spawn a few days after the full moon. But this specific day can be different, slightly different between species, and the exact hour is also slightly different between species. Here on this graph is an example for the croppers in the Great Barrier Reef that are known to spawn 1 to 4 hours after dusk, between 1 and 8 nights after the full moon. But most species actually between the nights 4, 5, and 6 after the full moon, and it's almost as a clock. It's just seems to be very, very well regulated. And I guess the big breakthrough was done by Jenny Craigs, that works at the Ornament Museum in London. And what he did in captivity was he tried to imitate these exact same cycles, the temperature cycles, the irradiance, the solar irradiance of the amount of light from the sun, how it changes throughout the year, try to imitate that in an aquarium. He also tried to imitate the irradiance of the moon phases, and also having the lights turn on and off to imitate the sunrise and sunset at the correct times that they would, as they would do in the field, on the reef. And obviously that changes from day to day, so that has to be all programmed like that. And also the same thing for the moonrise and moonset. And with all of this, it was actually successful to get the corals to spawn at the same day and same hour as they would spawn on a reef. He had 2 sets of aquariums. One of them was imitating the Australian regime, and the other one was imitating the Singapore regime, and he was able to get them to spawn at the exact same time, at the exact day and time as they would spawn in the field. So pretty cool. So we wanted to do that here too. So we've been trying to induce guided maturation and spawning of coral species from Florida in the lab. And to do that, we need to also do the same thing, mimicking the natural temperature, the annual temperature cycle, the sun and moon-like cycles, and then heavily feeding these corals. And this is very, very important in the Florida Reef Tract, because as you know, we are in a much worse condition than the Great Barrier Reef, for instance, which is not good, but it's still in a much better condition than we are here. So we do need to have restoration, almost even to prevent the loss to be as fast. And hopefully by the time we get around to controlling our greenhouse gas emissions, that we will still have some corals left to repopulate the reefs. So the first group that started this was actually the Florida Aquarium, which has done a fantastic job working with some of the most endangered species in our area. Here at Nova Southeastern University, we started working with the Montastra cavernosa, which is known as the Great Star Coral. So this coral is very important in our region, because I don't know if you're a diver, you have seen this all over the place. In fact, this coral species accounts for about 50% of the coral cover in our area. So it's a very important coral, and it's been very affected by coral disease lately. So their numbers have been going down a lot. So we definitely wanted to be able to get to reproduce this coral in captivity and learn more about how to reared in captivity, and then at some point hopefully be able to put it back on a reef. So we set up a system kind of following, Jamie Craig's instructions. So we have the two different types of lights. One of them, so these are the radiant LED lights that imitate the full spectrum of light. So they're imitating the sunlight. And then we have this other lights here that imitate the moonlight. And then this system has 2 raceways, and then the water just needs to go through, like a filtration system, UV sterilizers to remove any bacteria and things like that. Protein skimmer that removes proteins and lipids, out of products of their own. After feeding, they release stuff in the water that needs to be taken out of the water. Bioballs that kind of remove ammonia that is toxic for the corals. And then calcium reactors to keep the alkalinity as equal as you would have in the ocean so that they can continue to build their skeletons. And then he has this black curtains around here that are just open for you to see what's inside, but they need to be completely covered, with this opaque sheet so that it doesn't have any interference from outside light into their natural cycles. To create this, as I mentioned, to imitate the solar irradiance, we use these lights, the Ecotech lights, radiant lights, ProGen 4, that can be controlled using this Neptune Apex system, which is like a computer that just tells the lights when they should be turned on and off, and what times, and how the light should start increasing from sunrise to about midday, and then start decreasing as sun sets. But we make it follow the natural solar irradiance. So we got data from NASA, and we've used their curve to imitate the natural light levels that you would find on a reef. So it's actually about minimum of 140 micromoles per square meter per second in the winter, and up to 250 in the summer. And then we program it with a actual sunrise and sunset times from Florida, from our region. In terms of the lunar cycle, the same thing. We explain when is the full moon, at what time and what hour, and then it goes and it programs it so that the moon rises and sets at the times that it would do. It actually doesn't outside in the field. And this lights produce very little amount of light. So the maximum light that they produce is just about 1 lux, and this is during full moon. And then on top of this light, actually, we usually put in, this is the light, this little thing here. We actually put a ping pong ball, actually, to just diffuse that light even more. We program it with a moonrise and moonset lights from our area too. And then we use, obviously, heaters and chillers to keep the temperature at the levels we want. And the temperatures we want are the natural average temperatures throughout the year here. So we went used data from one of the other labs here at Nova Southeastern University and measured the temperature on the reef every day for 11 years. And then we made an average of all those temperatures. We removed years where there was bleaching occurs because those temperatures were already stressful. So we removed those years to make this, to build this curve. And then we just make our systems imitate this curve throughout the year. And then we feed them heavily. This is the products we've been using. And I'm showing you here because they also, I think it's a lot of you that have aquariums are always interested in this stuff. So we use products from Polyplab, Reefroids and Colors, and then from Reef Nutrition, Oyster Feast, and Real Oceanic Eggs. Those usually have very high content in lipids and fatty acids, which are essential to create eggs. And we feed them about 6 days per week. So what is known about when is it that this coral, the Montasher cavernosa, that was the one we were interested in reproducing in captivity, when is it that is known to spawn? Throughout the Caribbean, is known to spawn 5 to 10 days after the full moon of August or September, about 15 to 165 minutes after sunset. So that's what's known about this species throughout the Caribbean, so for all the different regions. But for our area here in Broward, my lab has been collecting data also. And so, for instance, in 2015, we observed them spawning 6 days after the full moon, from 6 days to 10 days after the full moon, with more of the colonies spawning around 8 days after the full moon. And this is the exact time they did so, about 70-something, 50-something minutes after sunset. This is when we observed them spawning. And in 2019, the same thing, we were also observing it, and they also spawned mostly this day 7 and 8 and 9 after the full moon. So that's, and again, between 108 to 103 minutes after sunset. So this is very, you can see that this is just very, very well regulated, and it happens always at the same time. And so it is very, very interesting how it's so well synchronized. And is the times are well within the programmed, I guess the values that have been found for other places in the Caribbean. So because of that, our expected spawning time, our due date for our corals, was August 8th to 13 this year, between 8:20 and 9:50, and then September 7 to 11, between 7:45 and 9:15. So that was our estimates based on everything that we knew for these corals. And we did get to see corals spawning. So what you see here, this is a coral in our lab spawning, and you can see this is a male. You're going to start seeing on this side, and it's pretty impressive. So it's like a cloud, almost looks like a cloud of smoke that they release. It's very interesting. Before they release, these polyps just get very tall, like a tower, and then they just compress and release the sperm. It's pretty unique. By this, whenever this happens, everybody in the lab is just very happy. It's been a work of a year. I'm going to just show you a little bit again, but it is pretty cool. And it can happen that it's about just one region of the coral that releases. Usually each coral, the males release about, sometimes every 3 minutes, every 5 minutes they release, and they do it probably for about half hour to an hour. It depends. And then the females do it too. So this is a female here. Just so just take a look and see how it's a little bit, well, it's similar, but you see how they look also like a tower. And then these ones, you definitely will see them contracting. Here we go. So pretty cool. I mean, when this happens, we are all very, very excited. And again, I'm just showing you a video of one of them going, but this is happening, throughout the tank at, with 5 minutes, 10 minutes differences from each other. Females usually release eggs about once or twice per night, with the difference maybe about 10, 15 minutes from each time, while males keep on releasing over and over again. I'm just going to show you one more time just because I like it. Yeah, so it is really cool. Interesting also, the snails that you see on the right side sometimes also decide to spawn on the same nights as the corals, which sometimes can be problematic because you get then the sperm and the eggs of all these two species together. Anyway, the eggs have a lot of lipids, so they all float to the surface. But at this time, the tank is full of sperm, so as the eggs are released, they go through the cloud of sperm and get fertilized that way. All right, so how does that compare to what we expected? So this is what we remember. In blue is what we had expected. What we actually observed was that, so we expected them to spawn between days 8 and 13 at between 8:20 and 9:50. We saw them going nights 8 to 12, between 8:28 and 10:31. So that's actually pretty good. September 7 to 11 was our prediction, between 7:45 and 9:15. We actually saw them spawning 1 day earlier than we were expecting, and night 6, 7, and 8 at between 8:30 and 9:54, so a little bit later year. The most of the corals did spawn in August, so we think the September was more of just a leftover. So again, we were able to see them spawning at the same times as they would spawn in the field. So it's pretty cool that we can replicate these natural cycles in captivity. And we got a lot of eggs from this corals, like thousands and thousands of eggs. We got very good some nights we would get very, very good fertilization, particularly if we had a lot of males and females spawning at the same time, because the more males and females spawning, the more there is compatibility between them. You can have, I guess, when you have two individuals that are more related to each other, their fertilization rates are not as strong. So the more corals you have spawning on the same night, the more you will have chances of having higher fertilization on your cultures. So we got that, and we got a lot of babies. And the next day would come in, and you would see this well, embryos that started like this to actually look a bit like prawn ship phase, or we sometimes call it cornflake stage. They look very different the next day. This is just about 12 hours afterwards. And then after they've done the fertilization, we need to remove the sperm out of the water, because if we put this water with all the sperm in our cultures to rear the embryos, the sperm will just die and degrade the water. So we need to get rid of the sperm. So the way we do it is with the good old gravy separator that a lot of you probably use for Thanksgiving. What happens is that the eggs, as you can see here, have a lot of lipids, so they actually stay at the surface, while the water with sperm stays throughout. So you can use then gravy separators to remove this water with the sperm, and you can put more water in, mix in the eggs, kind of wash them a little bit, let them come again to the surface, and then remove again the water with the leftover sperm. And then you have particularly really clean embryos that you can use for culture. We've been culturing them in these tanks. So these tanks have been funded through the Hoover Foundation and the Friends of Our Florida Reef. So this is a mass scale larval rearing system for corals. The good thing is it allows us to have different larvae from different species in each tank, and actually have batches from different days in each tank also. And so we can rear thousands and thousands of larvae in each of these tanks without having to wash them, so which is a very good system. So after, depending on the species, after about a week or just 3 or 4 days, this larvae actually, the embryos have developed into a larva, and then they can be settled. So I'm just showing you how the larvae swim around. So this is coral larvae. I know people think larvae are always very disgusting, but actually I think they're pretty cool. I think they're fun. So this is the stage that they're looking around and poking the bottom and seeing if this is a good spot to settle or not. And when they are, we put them with this ceramic tiles, and then they actually attach to these tiles. And what you see here is already these little pink things, actually all settlers. This is a really high density of settlers. We didn't think that they were going to settle this much. So we actually got a very, very high settlement rate, more than we even wanted. We probably, if we knew, we'd have to give them more space for them to settle. But what happens is they settle on this tiles, and then they undergo a metamorphosis into a polyp, and then after a few days they develop the tentacles and become like that little miniature of a coral, and then that's the one that polyp will grow, and then will start making copies of themselves to become a colony. So that's just the whole process. So just to show you how they look like, this is a 1-week-old coral, already has a little bit of those little symbiotic algae. So most of the species do not have the algae in their eggs, so they need to then acquire it from the water. So this one has already acquired a few, and this is about 8 to 9 weeks. You already see a lot of the algae. All these little dots is a little algae living on them. You can see already a bit of tentacles, and this is their mouth. And this is just showing you how they look like. We've reared these corals in the past. It's about a 1.5 year Montasteric cavernosa, and this is about a 3 years old Montasteric cavernosa. So they grow slowly, corals grow slowly, and that's one of the problems, because if we are to grow them all in captivity until they reach a size that they're good to put out on a reef, and that they're less susceptible to predation and less susceptible to environmental change, it takes a long time. So another thing that I will talk to you a little bit about in the end is about ways we're trying to figure out how fast can we put them out, so that we don't have to keep them in laboratory for such a long time, which is quite a lot of time consuming and expensive. We also I want to show you, because we've been rearing larvae of other corals, some of them that we've been rearing in captivity, other ones that we receive larvae from other places. These ones were not induced to spawn in captivity. Well, actually, these ones were collected right before they were, particularly this one, staghorn coral, was collected right before the time that they were supposed to spawn. So they already were pregnant and they were all into the queue. So we just put them in the right light, the sun and moonlight, and then they also spawn at the right time as they would on a reef. But they were not kept in captivity for a whole year as the corals that we had before. So these ones have, this is just a true development, and a little polyp here already with some tentacles. Again, as they start developing another little polyp, you see here they already have the original polyp already has budded and has now 2 new polyps. And this is just how they progress into the stages and start having more and more polyps. And so this is actually really, croppers are known to be fast growers. So 16 weeks they already have a lot of polyps. Agaricea is a coral that you see a little bit in this area, a lot actually in the jacks, for instance. You see Agaricea a lot. It likes hard substrate, but is the brooder. Is actually pretty easy to rear in captivity and makes this beautiful polyps, and it's so it's a very easy one to rear in captivity. Pyxostreoides, a lot of you have seen it. This is known as the mustard eel coral because of this mustardy coral. But it's funny, in our region, it doesn't have this color as much. It has more of a brown morph, but it has exactly this shape. So if you've been on a reef, you've seen this coral for sure. Their babies also grow very fast and have very interesting colors. So sometimes the mouths are green and the rest is brown. It's kind of an interesting type of coral. I also wanted to show you how the babies are very different from species to species. So it's another interesting thing. And we've been trying is to establish the best way to rear this baby. So most of the time, people would, and if maybe 10 years ago, we would rear the larvae, do some experiments about settlement, but then we would not be able to keep these corals alive. And now, more recently, we've been optimizing the ways to keep these babies alive, which is hard. And a lot of it actually has to do with the fact that the conditions that make this coral survive in captivity are a bit different than the ones from adults. The difference is they do not like light. On a reef, usually babies settle on crevices underneath rocks and things like that. So places that not have a lot of light, and there's a reason for it. And actually, when we do not expose them to very high light right from the beginning, they have some time to acquire their algae that allows them to then do photosynthesis. And only after they get to a certain size and have tentacles that they can eat from the plankton, as I'll show you a little video afterwards, then they usually then can start withstanding a little bit higher light or the same light levels that the adults do. But the mountainous star coral that you see here a lot in our region too, usually you don't see a lot of small ones. You see a lot of, sometimes there's sites that just have these very, very large ones, about 2 meters big. They're really, really large. This is a coral that is very, very susceptible to disease. So his numbers have been decreasing dramatically in our area. We've been able to rear them also. Again, this is 3 weeks old, already has a few, each of these little dots is algae, is unicellular algae that lives on their tissue. And as they start growing, they start getting more and more of this algae inside, and then they develop nice polyps, and it's just a very pretty coral. Copophilia natans, so these ones were actually, we've been able to rear these ones from larvae that were spawned in captivity, like the way we did our Montastra cavernosa, but at the Florida aquarium. So they actually had so many larvae. They got such big production of babies that they couldn't even handle all of them. So they actually have given us some larvae of this species and some other ones that I'll show you afterwards. So we've been rearing their babies. So this is here a baby Copophilia natans. It's about 6 weeks old. We also receive larvae from them from the symmetrical brain coral, Pseudodiploria strigosa. So this is the little babies, very, very small here, about 6 weeks old too. And another one that we're very happy to have is this one, the Dendrogyra cylindris. And I'm thinking a lot of you have never seen this here on a reef because it's really, really rare. It's now actually already locally extinct on the northern side of the Florida Reef Tract. And there's some, like in the dry tortugas and very, very the southern part of the Keys and very few numbers because they're very, very susceptible to this disease that has been killing a lot of corals in our region. So we actually, the Florida Aquarium received some of the pieces of this corals, the ones that were, the disease was just going through them and killing them. So what they did was just they cut a part of this coral that was still alive and it was just going to die in 2 or 3 days. And they brought it to a lab and they've been keeping them, and now actually they've been able to get them to reproduce in captivity. So now we're rearing the babies of this species. And actually now we have tons of them, which are much more than exist in wild. In the wild, there's like 20, 60 or something of this colonies. And now we have hundreds of thousands of these corals in captivity. So now we have a bigger population in captivity than there is in the wild, which is really, it's good news and bad news, right? So this is this is a 6-week-old baby, and this is a 2.5 months old. They actually are doing pretty well, and they're eating, and they have large tentacles. So we're hopeful that we'll be able to grow them to a point that we then can put them back on a reef and epoxy them back on a reef, and particularly if the conditions, obviously, and the disease abate. Diploria labyrinthiformis, this is the groove brain coral. We also have been able to get larvae from this species. Been rearing them here. Again, in the beginning, they don't even have tentacles. This is a 5 days old one. Doesn't have tentacles, doesn't have any algae on them. And then as it grows, it starts getting more of that algae, larger tentacles. And when they have these large tentacles, then they start eating very well. This is a 10-week-old coral, beautiful. And after a certain point, they actually start creating this bud. So this copies of themselves. This is a clone of the original polyp. Here, say this one already has 2 clones. And then obviously as this polyp grows and becomes the same size as the original polyp, then they'll start heading and becoming a bigger and bigger colony. And I want to show you this video because this is a video of a coral, a baby coral eating. And so this is here an egg of another animal that is going to be eating. So this is their mouth. And you'll see they will push this into their mouth and then it will swallow it. This is obviously in fast speed. So put it on his mouth, and it will now start opening its mouth enough that it can just swallow the food. And that's it, gone. So they eat very well, and in captivity, they have been, if they're in good conditions, they're well fed, they tend to grow relatively fast too. So that's very good news. Oops. All right, so we've been growing these corals in the lab, and this is the part that we have been trying to optimize now, which is how to grow these different species in captivity and how to optimize the amount of light they get and the food that they like and all these things. But we've been rearing them in tanks indoors, but also outdoor tanks. Indoors, usually more in the beginning when they cannot withstand such high amounts of light. And then when they get a bit more, a little bit bigger size, then they're moved to the outdoor systems where they can deal with higher light levels, and which they do like. They like the natural sunlight has some properties, and the lights we have in captivity are still not as good as. So they grow better outside, but only if they're a certain age and at a certain development stage. What we are also trying to figure out is when can we start putting them growing out on the reef and gluing them back into the reef to start repopulating areas that have been disturbed and therefore need to be recovered. So what we grow them in this coral trees that are also used to propagate corals through asexual reproduction, so through fragmentation processes. And what you see here actually on this tree is like what we call kebabs of tiles, and these tiles will have little babies on them of different ages. We're putting them out at different ages and trying to figure out at what age is it okay to put them out. So and so we check their survival and their growth over time and try to figure out, can we put them out right after settlement? Can we put them out after a month? Can we do it 2 months or 3 months? Because the faster we can put them out on a reef, the less work and the less time we have to work on them, and the cost of rearing them gets decreased a lot. So that's what we want to do. And then the next stage, obviously, after this is to try to actually go and glue them back on a reef and see them growing. So just these names are just, we're scientists, but we're fun. So we like to give the coral trees funny names. So this is the Morgan tree and the tree rex. They all have different names like this. And I just wanted to finalize by obviously thanking my lab. This is my students. this is the students that work in my lab, Morgan Short, Murphy McDonald, Ashley Steinberg, Rachel Yonata, and not pictured here, Michael Hu, that has just joined the lab recently. And I wanted to also finalize by thanking the agencies that have supported our research. So we get some of our funding through the Protect Our Reefs license plate initiative that is promoted by Moat Marine Lab. We've received a lot of larvae from the Florida Aquarium and share a lot of information with them. When we got our Montasha cavernosa, we also got a lot of eggs and larvae, so we also gave them some. So when they have a lot of extra larvae, they gave us some. So we're all trying to learn, because the more labs that are trying to figure out the best way to best recipe to grow this coral is the best. Need to thank also the Florida Department of Environmental Protection, which funds most of our research. The Florida Fish and Wildlife that gives us the permits to work on this. As I mentioned, the Friends of our Florida Reef and the Uber Foundation that have funded our large-scale, mass-scale larval culture system. And obviously NSU that supports all our work and our students. So thank you very much for listening. And if you have any questions, I'll be happy to answer.
Okay, wonderful. I'm going to stop this screen share so that we can see everything happening. And perfect. Yep. There we go. Excellent. Okay, we had a number of very interesting questions, and that was a very fascinating presentation. That was fantastic. Okay, so one of the fun ones, can you elaborate a little bit more on what makes the spawning process smelly?
Oh, I guess is the lipids, and that is just it smells like fish, really. You know, like if you go to fish market, that's kind of how it smells like. In reefs that there's so many corals and they're all spawning at the same time, there's all this fish material, like fishy lipids material, kind of a mix between degrading and still good. So you can actually from shore smell that they're spawning. Oh, wow. I mean, I have never smelled it here in Florida because we have a smaller coral cover, but in Okinawa, definitely I remember smelling it. Definitely, yeah.
That's really interesting. Okay, so obviously there are a number of factors that contribute to the spawning process. Is there any indication that once a coral begins to release eggs or sperm that's a specific trigger to all of the other corals in the area to release as well, or is it just the overarching factors?
There's some research that seems to we haven't been able to identify the pheromone that they might release, but there's some evidence that may be possible, but it hasn't been clearly identified. And there's some people that have been trying to try to find exactly that pheromone and when it's released and if there's something in the water. But we do know that when they're together, they usually synchronize it better than when they're separate. But we do not know if what's causing that is because they are at slightly different environmental conditions. So we cannot be sure. Scientists, we cannot tell you confidently that happens.
Okay, very interesting. What is the success rate of a coral to make it to adulthood out in the wild?
Ooh, I don't actually. I think there's a study, like generally for marine invertebrates, they think about 95% of them die. In a year, probably 30% or 40% maybe will make it, but then the most of the mortality actually happens on the first few days when they're very small and very easy to be predated. And then as they get bigger, they can start being less susceptible to predation and less susceptible to competition. Because what happens though is if they settle on a reef, this is a good spot, but then an algae starts growing on next to them, and the algae always grow very fast, much faster than corals, so they can overshadow them, and just by blocking their light access, they will kill the coral. So the probability that a coral will reach adult in the wild, it's very low. But again, they produce thousands and thousands and thousands of eggs. So that's the whole process. The same thing with fish, right? They have produced thousands of eggs, and maybe one will make it to adulthood and to reproduction size. So that's what that's what would happen in corals. Obviously, it's very different if a reef is pristine or if it is in an area that has a lot of pollution. and or in an area, area that is very polluted and there's a lot of sediment on the bottom, like there's a lot of dredging happening and there's always sediment, it's even hard for larvae to find a place where they would settle. So there might even be that extra problem.
Okay, interesting. Super quickly, how does the wild success rate compare to that in the lab that you guys have seen so far?
We've been improving it. No, with time, we've been getting better and better. So the success rate is still not what we would like, but it's even when we have like 10% success, which actually now is getting better than that, definitely. It's still much better than we would get in the wild. With the success that we have at the moment with some species can be really high as 95% survival. They're just perfectly fine. Other ones have about maybe 50% mortality right in the beginning, but then they actually don't die after that. So it depends on the species, but it's getting better and better every year. The more time we do it, the more labs that are working on doing this work, the more we're getting the right recipe for each species.
Okay, interesting. What size are the coral larvae before they attach to a structure?
It depends on the species, but the biggest ones are only 500 microns. Okay. So half a millimeter. So it's very, very small.
Teeny tiny. And then we had a question asking, are the coral larvae and gametes the same?
No, so the gametes is the eggs and the sperm, right? Then the eggs and the sperm, the sperm fertilizes the egg, and then the egg becomes an embryo, right? And then the embryo goes through gastrulation, and then it becomes a larva.
Okay, interesting. How old are corals before they're
reproductive?
Yeah.
So it depends again from species to species. Some species can become reproductive in about 3 years to 5 years. Other ones can take a little bit longer. It's more, it seems to be also a question a bit of their size. So the species that grow faster tend to reach maturity, sexual maturity earlier on, 3 to 5 years. Other ones can take 10 years. So it depends. And again, it will also depend on the quantity of food available in the area, if they have to fight other stresses, and if have to put their energies in towards fighting other things instead of putting their energy into reproduction.
Okay, interesting. So there were a couple questions. Do we know of any outside factors, things like vibrations, that may not naturally occur, but that may help induce coral spawning? Or is anyone researching any outside influencers that we can put into place to actually have corals spawning more than once per year?
No, none of that has been shown in terms of vibration. In terms of trying to get them to spawn more than once, because again, the egg takes 9 to 11 months to form, so that's not a lot of time and then they need 1 or 2 months to kind of recover from all the energy that they expended during the spawning. What we are thinking though is that the eggs, so when they lay the eggs in the end of the fall, the egg doesn't mature too much during the winter months, and then once spring hits, it starts actually maturing more. What some groups, and I'm going to be doing that this year, is going to look into potentially reducing the winter season and trying to have them, so just making a shorter winter instead of 3 or 4 months, making like a 1 month thing, and then start increasing right away the temperature and see if they can do it. But if we can do that, probably will be reduction from, instead of a 12-month cycle to maybe a 9-month cycle. We probably cannot reduce much more, because if we increase the temperature too rapidly, we will also not give them the cues that they are known to react to. Again, this is what our current knowledge. This is what we're trying now. Maybe if that works, maybe we'll try to go even further, but that's what we're trying to do at the moment.
Okay, interesting. What's being done to stop the spread of diseases among corals?
What is being done to stop the spread?
Stop the spread.
It's very difficult to stop the spread, because what we think is happening is that these environmental conditions are now so bad. There's so much pollution. There's so much stress with temperatures and other things happening, that the immunity system of the coral is very compromised. So it just becomes much more vulnerable to pathogens. So we think the pathogen is probably more of a secondary thing. It's just that the immunity system of the coral is reduced, and therefore then is more susceptible to pathogen attack. What is being done? Very little. Well, a lot of research, but not in the field. Very little, because we just don't even know what we can do. What is being done in some areas that is having areas. So when they see a coral that is diseased, has a disease in one spot, what they do sometimes is then they use an antibiotic, an epoxy with an antibiotic that is applied to those areas that have disease. And now they've tried different products, and now they actually come up with some that are more effective and that actually can cure this disease on this spot. But sometimes then it appears in another part of the colony. So it's a good it may be a good methodology to save iconic reefs and reefs, very old corals and things like that. But as an overall large strategy for the reef track, or for the whole Caribbean, because it's spreading to the Caribbean, it's not a good strategy. But in terms of research, there's now some research about using probiotics to try to increase the immunity system of the corals and try to get them to be more resistant to this disease.
Okay. So where can we follow your research online? Any online research resources where we can see what you guys are doing and progress being made and things like that?
We have a website, and we have also the YouTube channel that I provided the link. I think you're going to be providing the link where we put down some of the videos of what we're doing. We have a Twitter account for the Marine Larval Ecology Lab that you can follow also there. Actually, probably that's the most, the one you will see the news coming on a more regular basis.
Okay, excellent. I will, to everybody who's tuning in, I did post a link to YouTube where you can actually see some of the videos that we saw tonight, and I'll go ahead and I'll get information and social media handles, and then when we post the recording of the talk, I'll be sure to include that information in our description so that you guys can go online and you can continue following and seeing additional updates. And then last question, I think, for the night, how can we actually support coral reef restoration and research efforts?
I guess this is a complicated question because there's a lot of levels. Starting by voting on people that support environmental measures, that's one of the things, obviously, because that will increase, bring awareness to other people that are not divers, that are not aware of that what's happening to the reefs and how important they are for even for our coastlines and for our protection against flooding. So that all of that is important. And then in terms of doing things yourself or making donations to agencies that do restoration, so the Coral Restoration Foundation. Here at Nova Southeastern University, we have the Coral Nursery Initiative that also receives funds from donations for the research and for the propagation of corals and outplanting of corals on the reef in our region. So there's some options. There's some internships that you can do also at the CRF, and the Keys also have internships. But I think above all, what we can all do is bring awareness to people, and then honestly voting on the people that support environmental protection. That's the best you can do, and probably the most effective thing you can do.
Okay, excellent. That's always helpful. And we've worked with a number of different presenters to try and help guide people in understanding how they can contact their local legislators and get a little bit more involved in just the lawmaking process in general, so that the interests of our divers are actually seen in the policies that are being made. That was a very, very informative presentation. Thank you so much. I did let everybody know that the YouTube link is posted. If you guys have additional questions, you are more than welcome to email me. It's marketing@puravitadivers.com, and I'll pass those questions along. And then Rachel, it looks like just posted on Twitter. It's @NSU Marine Larval. And then again, we'll include all of those handles so that you guys can follow Dr. Joana's research over the next few months and years. Just a quick reminder, we do have our coral reef conservation class coming up on the 27th at 6:30. So it's Patty's specialty. If you guys would like to join that, feel free to email me or you can sign up online. And then just thanks everybody for attending. Thanks, Dr. Joanna. That was fantastic.
Thank you.
Excellent. So everybody get home safe. Have a great night. And we'll have the recording posted on social media over the weekend. So keep an eye out for that.
All right. Thank you so much.
Thank you.